All right, let's see. Come on, share screen. There we go. This one. All right. So first of all, you guys saw the schedule. I had the schedule up there. I'm only going to see you guys in lecture 3 more times in person. That's crazy talk. We're going to. I have for you on online Friday. I put up a lecture on the first lecture on the nervous system. So that's up there. And last week there was we took the exam and there was nothing. Well, I put up Friday, but there's nothing there. I have 6 lectures for you. And I'm getting 1 here, 1's online, and then we have the next 2 weeks, I'll have 1 recorded and 1 in person. So if you're looking ahead, there's going to be 6 lectures for you on chapters 10 and 11. So big ones, the nervous system, like I say, it's in that first lecture, it's one of the difficult ones to learn and to teach. You know, the brain is, if you read my first lecture, I go into a lot of things talking about this is the final frontier, how little we know about the mind, you know. And today I'll talk about the physiology of things, and we'll see how far I get. I may delve into the beginning of the anatomy. Look at the time. All right, so you guys have to know, lab will be practical this week or next for you guys. And then I put up a lab current cloud for you guys to do in the next 2 weeks on the nervous system. So we'll do that. There's a 3-parter, so take your time. You can do 1 and then come back to it, and then there's a quiz at the end. So there's the current cloud goes through the central nervous system, the brain, and the spinal cord. So it's helpful for lecture and for the practical. So I'll have the brain and the spinal cord, and then next week we'll have after the next lab will be on the senses. We'll do eye tests and maybe look at reflexes. So that's the last labs will be on senses and reflexes. After this 2-week block where we do brain, spinal. That's it. The last practical test will be online. And before you go to Thanksgiving, we'll have one more exam in here on the nervous system, chapters 10 and 11. Any questions there? Guys, just learn this 10 and 11, the nervous system. Watch my lecture. It's a 2-parter on the nervous system in general. All right. Cool. All right, well, this nerve is amazing that it can produce electricity. I know, just like your battery. So here is a neuron. And amazingly, I think you guys know, it produces electricity. It's going to travel down that axon, which can be a meter long, go all the way down from your spinal cord in your butt region, all the way down to your toes. And you wonder, how can you make electricity in your body? That seems weird. I thought batteries did that, things like that. Well, we can do it using ions that are charged. Sodium is positively charged, chlorine is negatively charged. And let me just tell you, this difference in the charge is the voltage. The car's got a 12-volt battery. The laser pointer has a 1 point. I don't know what it is. A very small battery, and that voltage difference can do work. So the bigger the voltage difference, the more work, the more dangerous it is to go through your body, right? And this plug, if there was a plug in the wall, that electricity is just electrons that are flowing, and they need to flow. So you need a circuit. You got to plug it in as 2 prongs, right? And as long as it's grounded, electricity will flow, and it can do work. You can run a fan, a light bulb, all these things. So that's the deal. I'm going to get the physics of it. But you can produce electricity in living cells by having a difference in like sodiums and potassiums and chlorides, things that are charged. So that's the way we do it. And all of these billions of neurons, let's see, 100 billion neurons in your brain, they're all constantly using ATP energy to keep this voltage difference between the inside and the outside. You see, they're more negative inside, like negative 70 millivolts, more negative. And they're using energy to do this pump, to pump sodium and potassium to make this difference so that they're always ready to fire. So this axon, this neuron can fire, and it can fire 100 times a second, like, or just one time a second, whatever, you know? And so it's ready to go. It changes this membrane potential, the difference in electricity. So it's pretty impressive. I am impressed. All right, so how's that sound? A cell membrane. Yeah, I'll show you exactly what that looks like. So if you did this experiment right here, and we can do this, we're not going to do this, but we can do this, where you take out a single nerve, and there's giant axons in squid, lobsters have these big axons, and so we don't care about killing squid too much. It's not like a dog or something, right? And so we take out the nerve, this big long nerve, we put it into saline solution, and then we can fool with it, and we can put a little tiny probe in there and measure the electrical difference between the inside of the axon and the outside. And as you can see, when it's on the outside, you got nothing. Here's a pen. Yeah, I can draw. Yeah, so nothing. But then as soon as you push that one of the probes inside, whoop, the electricity, it just jumps down to -70. Like, damn, snap, that's awesome. You can see that difference there. And so as long as the axon's alive, it keeps that voltage difference. And you pull it out again, whoop, it goes back to 0. So anyway, you can really measure this easily inside your neurons. They're really doing this. And you just have to memorize this number. Inside your nerves is -70 millivolts. The 12-volt battery, millivolts are thousands of yes. It's a very small voltage difference compared to, the batteries you guys use in your electronics. That's okay, because this millivolt difference is not doing any real work. It just uses a signal. It's going to fire. It's going to be a signal. So like I say, it's not powering a fan or a microwave. It's just a little blip of a signal. Is all you need. All right. So how do we get there? So I'm going to tell you this. It's a little confusing, but I don't think so. I think I'm going to break it down very simply for you guys. So this difference has to do with, you need to memorize this as well. When you look at any cell, really, especially look at a neuron, you've got tons of potassium ions inside, and then more sodium ions outside. Now, if you have more positives outside than inside, it's going to be relatively more negative on the inside. So if you throw more positives out, it's going to be more negative on the inside. And you just got to know this, all cells, potassium is high concentration intracellular, and sodium is more concentrated outside. So you have this gradient, this difference. And you all know, because of diffusion, what wants to happen? That sodium wants to diffuse in, right? Because everything goes from high to low concentration, right? Simply. And that potassium wants to diffuse out. So we're going to see, they leak a little bit, but your cell works hard to keep that difference, keep the sodiums out, keep the potassiums in. Yeah, and we have this pump. It's showing the pump up there going. It takes energy, ATP. We're constantly pumping out 3 sodium for every 2 potassium that come in. And after a little bit of time, it's going to get more negative inside, isn't it? Because if you kick out 3 positives for every 2 positives that come in, it's going to be negative just in a short amount of time, right? All right, so importantly, these are the 3 things. These are the 3 things of the factors that are going to be involved. Well, first is diffusion. Everything wants to go from high to low concentration. Right, everything. So that sodium on the outside, it would love to be inside, man. There's not much sodium in there, and it's like, dude, there's no sodiums in here. I could like stretch out. So it really wants to get in there. And the potassium wants to get out. I mean, if you just look at purely diffusion. Let's look at a couple other things too. Charge. So the thing is that opposites attract. And so that potassium on the inside is positively charged. And there's more of it on the inside. So diffusion tells me it wants to go out where there's less sodium, with less potassium. There's more inside. But the charge means if it's more positive outside the cell, and I'm positive, I don't like positives. Like 2 magnets, you try to put them together with the wrong poles, they repel each other. So charge is going to be important. Things are going to go towards the opposite charge. So imagine this, it's more negative inside the axon, and all of a sudden I open up these gates, all these positively charged sodiums are gonna rush in, because damn, it's like the magnets snapping together. So charge, things are gonna go, not tend to go towards the same charge, and towards the opposite charge. And then lastly, we gotta look at this membrane and see what is it permeable to, what can go through it. And we'll see sodium and potassium, there are channels that they can leak through at a certain rate. And sometimes you can close channels, open channels. And then very importantly for our story is that there's a sodium potassium pump. It's a specialized protein that's gonna pump out 3 sodiums and pump into potassiums when you give it ATP. Whew. All right, so I got it. So diffusion, what concentration gradient is. The electrical charge is going to be important, which way it's going to go. And then the permeability, and are you actually pumping something? Are obviously going to make a big difference. That's not that one. Now we'll get to what's that. All right, so these are one of these channels right here. And there are channels along that axon that can make sodium come in and potassium go out. And so there is a little bit of leakage, but luckily this pump can overwhelm that. But there is a slow leak all the time, but the pump is going to overwhelm that. Yeah. And this is the important guy, the sodium potassium pump. It's in all your results. It uses a lot of your energy. You guys have breakfast, a lot of those calories go towards running these pumps. So they run 24/7. And they've got a configuration where they take 3 sodiums from the inside and they dump it to the outside. Then it changes shape and it's like sticky to potassium. It takes 2 potassium from the outside and then it changes shape and pumps it on the inside with ATP. So as long as you're giving it energy, it will do this pump. 3 sodiums out, 2 potassiums. And the net effect is you're going to have a lot more sodiums on the outside and a lot more potassium on the inside. And you're going to have a 3-to-2 ratio, meaning it's going to be more negative inside because you kicked out 3 positives for every 2 negative. Yep, so this uses ATP. You guys have to be alive for this to work, and you need to always be making energy. And that's why your brain is so expensive. It gets so much glucose. If you guys have low glucose levels in your blood, pretty soon you're going to pass out. You're going to have issues with your brain because your brain is so hungry because it has to always be pumping this or else your neurons don't work. Neurons don't work, your breathing doesn't work, your heart doesn't work, you're dead. All right, here's a little 3-slide little montage here. So imagine you're a cell here. And the deal is that there are channels for both sodium and potassium. Like I said, they're leaking. They're leaking a little bit all the time. And actually, potassium can leak, is much leakier. I'm saying that the membrane is more permeable to potassium. So potassium is going to want to leak out. Sodium wants to get in, but it has a harder time. It has fewer channels to go in. So there's an overall potassium is going to leave more than sodium comes in. All right, bam. Then it's automatically going to make it more negative inside. Because if more positives can leave, then you're going to have a little more negativity inside. Just based on permeability. Secondly, let me throw this at you. Inside of any cell, there's a lot of negatively charged molecules. Proteins negatively charged, sulfates, nitrates, things like that. So there's overall these big negative molecules inside a cell. They can't leave, but they live in the cell. And so it's automatically a little bit negative because of some other stuff in there. So we have molecules that are negative in there. Plus we have more potassiums tend to leave. So more positives leave. Yeah, 2 things are going to set up this gradient automatically. We're not even putting any energy into it yet. Yeah. And that's that difference, that slight difference there, negative 70 millivolts. Cool. But there it is. There's the sodium potassium pump. And this sucker is hungry. It's using your ATP, right? You're burning sugar, you're getting ATP. And even though there's leakage, this pump is overwhelming it. All along the whole membrane, you got this pump. And it's pumping out the sodium, pumping in the potassium. And it's going to keep it at this negative 70. Yeah, if you were to die and run out of ATP, the pump would shut down, and then it would be more equilibrate. It would still be a little bit negative, but it wouldn't be -7. All right, a little bit of this, a little bit of chemistry. All right, you guys, so -70 millivolts inside, and because there's more positive ions on the outside of that membrane than inside, so it's going to be relatively more negative on the inside. All right, whew. Let's talk how neurons fire, right? So that's what we're going to get to here, definitely. We're going to see these neurons, this neuron that's up right in the board, it's all or nothing, man. It's not like half a fire. It's either going to fire or it's not. And whatever it fires, when that signal goes down, it's going to be the same size signal no matter what. So it's either all or nothing. And if it's all, it's going to fire a signal, an action potential, we call it. It's going to run down the end of this axis. That's the big picture. All right, so how does this work? That's what we're going to get to. This is how electricity is formed, and when the signal is a trigger, then when it decides to fire, and that signal is going to be carried down, it's electricity down to the end. This thing can be long, a long nerve. So it's going to go down the nerve. This wave is going to be carried down. That's the signal. I'll tell you how that works. What we need in this membrane are these channels that are going to open. And if you open up channels to sodium, sodium's going to rush in because it's positive, right? And the inside's negative. Hell yeah, I'm getting in there. Like a magnet's going to stick to it. Plus the concentration gradient. There's way more sodium on the outside, not much on the inside. Hell yeah, I'm going to come in. So if you open up the doors to sodium in this room, sodium would rush in because it's got a couple things going for it, the charge and the concentration. So these gates are closed, except we can open those gates. And then we can let them rush in. That's how it's going to change the polarity. It's going to change the electrical difference. So these gates, there's also some of the gates, I'll show you a picture next, but for your senses, there's light can hit your retina and it can turn on a neuron, make it excited, or pressure can do it, or chemicals can bind and do it, or electricity too. So here we go, let's look at them. So the first one is the important one for today's story. Today's story is voltage-gated channels. So that means when the electricity hits a certain point, this like tripwire, then they open. What we're going to see, normally this neuron is -70. I'll just give it to you. The trip is -55. If it's -55, that is the threshold, then it's going to fire. If it is -56, no way, I don't care, nothing happens. -57, don't care. -55, bam, it's going to fire. So these ones are gated and they're waiting for electricity to hit that trigger level and they open. If it's not there, they snap shut. So it's a voltage gated channel that's going to let the sodiums in. These other ones that let the potassiums out. And just to finish the story though, when you hear ligand, that's some kind of signaling molecule, a hormone, something. When the molecule binds to it, oh, this is also in the nerve too. This is like when the acetylcholine goes across the synapse and then it binds to the receptors. That's what's happening here. It binds to the receptors, it's going to open those channels, and then sodium is going to go in the muscle, and it's going to cause the muscle to contract. So ligand is some kind of signaling molecule, like that neurotransmitter. That's what's going to open the gates. It's going to be some kind of molecule. Voltage is electricity. And lastly, it just shows mechanically gated. I can't see this picture without thinking of 2 thumbs up. All I see. But yeah, something like touch receptors, you press on them, it causes those things to open and it fires. All right, so that's why we're talking about these gated channels, because that's how the signal is going to start. You're going to be opening these channels, and that sodium is going to be like, oh my God, the door's open. I love it. I can get in finally. Like some big concert or some sale, you know, it's Walmart opens at 2 a.m. or something. All right, so we're going to call this blip. It's going to travel down the action potential. And it's going to be super negative inside, right? It's going to be positive on the outside. And if we switch to that, and we're going to switch it, we're going to make it positive on the inside, negative on the outside. It's called depolarizing. Actually, bring it to 0, depolarize that. Ooh, yeah. So being polar, like having a north and south pole, means there's a difference. Depolarizing means getting rid of that difference. So if you get rid of that voltage difference, you depolarized it. And that's what's going to happen when a nerve is going to fire. Now, if I was going to make it more negative inside, it's called hyperpolarized. Like our political climate now is hyperpolarized. People are at two ends, you know. To depolarize it would bring us together. Now, meet in the middle and agree on it. That's pretty good, right? So in your nerve cell, it's usually negative on the inside. If you depolarize it, it's going to bring it down to zero difference. If you hyperpolarize it, instead of negative 70, it's like negative 90, negative 100. What do you guys think? I think I'm explaining it perfectly. I give myself an A. I like it. I think we're taking it stepwise, adding all these things together. Because like muscle contraction, how the nerves function, today's lecture can be confusing. So if you guys are a little lost, it's okay. Just try to keep up here, and then you'll be able to look at it yourself, and I'll be happy to explain it. All right, so the trigger for these voltage-gated channels, it has to hit a threshold. And we're going to see this nerve, remember it's negative 70 usually, negative 70 is the charge inside. If you stimulate it, you make it down to negative 60. See what I mean? Negative 60 is stimulating it. Negative 55 is going to be the trigger. So anything that's closer to zero is going to be depolarizing it, getting it, that direction. And so if you get to negative 60, negative 59, negative 58, negative 57, nothing happens. It's called subthreshold. You got to hit negative 55. That's going to be the trigger. And you do that by making the cell more negative, right? So open more sodium channels. I'm sorry, making it more positive, making it more towards the negative 55. So open sodium channels, sodium comes in, and it gets more and more depolarized. It gets closer to zero. And so this little illustration here shows you that these are sub-threshold. It didn't get to negative 55. Nothing happened. It was stimulated. Some sodium ions came in. It went from negative 70 to negative 69. And then it goes back to negative 70. But if you hit negative 55, that threshold, it fires. We're going to see what happens. Then it's going to fire. And you get one action potential, kind of like a muscle twitch, one action potential, and it goes back to normal. It's ready to be stimulated again. So this nerve can keep sending out signals, like I said, 100 per second, really fast. And I'll get to some other terms I see in here. One thing you might notice is that it's going to go up to like positive. Like you let in so many sodiums, it's completely flipped. Now all of a sudden it's positive inside instead of negative. And one thing you might notice too is that it comes back down again. It goes even in hyperpolarized. It's even more negative for a moment, then it goes back. Just notice that as well. All right, so this is what's happening. This is the neuron in question. This is the neuron that is going to decide whether it's going to fire or not. And if you think simplistically, my example I use in my online lecture the other night is that you're going to make a decision. This neuron's going to make a decision whether to fire or not. And it can listen to many people to make that decision. And so if people are egging you on to fire, they're egging you on, they're encouraging you, they're exciting you to go on, they're going to depolarize the membrane. They're going to open sodium channels, and they're going to make that negative 70 neuron negative 69, negative 68. They're going to start depolarizing it, bring it closer to zero. And one might not do it. Maybe two friends wouldn't do it. But if you have your whole family and all your friends telling you, do it, then it's going to bring it, so many channels will open that this neuron in question will get down to negative 55. And at that point, there's a trigger region, right at the beginning of the axon. And all the sodium channels, when it hits that trigger, negative 55, they open the doors. And then you're going to have this action potential. So the nerve itself in question is graded response. It can get more negative or more positive, but whether it fires or not is all or none. So on the flip side, you've got all your friends telling you to do it. But then you have a couple friends telling you not to do it. They've got all these reasons, and so other friends will be inhibiting it. They're going to be actually making it more negative inside. So now if you have equal numbers of friends telling you to do it and not do it, nothing's going to happen. So you need enough friends telling you to do it, excitatory signals, and then you're going to fire. Or you could have everyone saying, don't do it, all these negativity, this is going to hyperpolarize itself, bring it down to -80, and then you're going to fire no matter what. That's going to be really super polarized. So that's the deal. The neuron in question, it's great. It can go anywhere from -100 to -55, then it fires. So it'll go back and forth. It's listening to all these other neurons. And there could be 10,000 other neurons talking to it. By talking to it, I mean sending excitatory or inhibitory signals to be making it more negative or more positive. So that's the cool thing. You guys are in your decision making. It's neurons deciding to fire or not. They listen to all these other ones in these big circuits. If you guys make complicated decisions, it's hard to break it down to one neuron, of course. One neuron's not going to do anything, but all these neurons, whether they fire or not, it's going to come down to excitatory and inhibitory, and whether the nerve fires or not. You guys think? I'm giving you a lot. How long have I been talking? Oh yeah, it's about that time. I have a sense. Okay, I think I will just give you a couple more minutes. So, any questions here so far? You got it? It's normally -7. You know why, right? Because of that sodium potassium pump. It's more negative on the inside. And if you do depolarize it, it's going to get more towards 0. And if it hits -55, it's the trigger. And it's these special gates right here at the beginning of the axon. All the doors will open up. And then all the sodium is going to rush in. They're sodium gates. All right. I'll explain this and then we'll take a break and I'll come back and make sure you get it. So let's take a look. Look at this axon. Do you see the charge difference? I do. I see sodiums and potassiums. If you look at the figure, notice there's more sodiums out there, right? And potassiums. So it's going to be more negative on the inside because there's less positives. So I can see it's going to be -70 here as it's resting. We call it the resting potential, but that's a crappy way to describe it because it's not resting. It's constantly pumping. But that's where it sits, -70. That's the resting membrane potential. And if you hit -55, if that cell in question hits -55, right here, all these sodium channels, they're voltage gated, all the doors swing open. And all that sodium out there that's at the edge of the door is going to rush in. That's going to be the action potential. So all the sodium rushes in. If you look at the electricity inside the cell at that time, it's going to go from -70, -55. It's going to fire up not only to 0, but all the way up to +30. It's going to, all these sodiums come in. It's going to make it positive on the inside. Exactly the opposite of what it was like a millisecond ago. So that's depolarizing. And then polarizing again to positive. Now, this is the key. Those sodium channels snap shut. Those doors snap shut to sodium. All the potassium doors open up. And there's more potassium on the inside. Hell, it's not even more positive on the inside, right? So potassium wants to leave because of diffusion and because of charge. If you're potassium on the inside, and there's the outside, the doors are open, there's not that much potassium out there. So diffusion moves you out. And then it's actually more negative out there compared to the inside now. So you want to leave because you want to go towards that negativity. Oh, it's beautiful, huh? So sodium rushes in, doors shut. There's a delay. Potassium channels open. Potassium leaves. And that's going to, all that positivity leaving is going to bring it right back down to 0. Hell, it's going to bring it right down to negative, more than -70, down to -90. And then the cell evens it out back to -70 again. Whew. What do you guys think? And that is an action potential. It's a blip. And at the membrane of this thing, and it's just at the surface too, because in my mind, I thought, how the hell is there time for all the sodium to come in, and all the potassium to leave, and then to make it back to normal again? Turns out it's just at the surface. So you don't have to get the whole damn axon to change. Just at the surface, it's just like blips. Like I imagine like a little like your TV would change channels, like a little blip. That's all it needs is a little bit of the sodium potassium out. It makes a little bit of electricity, and that will propagate down the axon. And so you've got these trigger-fingered sodium channels that as soon as they open and shut, and potassium opens and shut, like delayed a little bit. If they all go at the same time, crap, it's not going to work. You need sodium open and closed, potassium open and closed. Yeah, and you get it. And so you get this little depolarization. And then you're going to see how it travels down the nerve. It goes down there. Let's take a look. And the important part of this trigger zone is this axon hillock. So the axon comes out of the neuron cell body right here. And these are the important ones right here. These are the ones that the trigger ones that they're waiting for that negative 55, and they're going to go. I'll give you guys the whole story here, actually. The neurons that come in like closer to the trigger zone have a little more input than the ones that come up further away. So like your close friends versus like your other friends you don't care about, you know, they're going to have like more of an effect too. So it's kind of cool too. If you're right by the trigger zone, you have a little more of an effect. It's going to change it in that area. If you're further away, it takes longer for it to get there. Yeah. Yeah, this is in words and what I've explained to you. You guys have a question? You can ask a question. So if you guys see this action potential, could you explain to me like what's going on when it begins? I can see it's at negative 70 is my resting. Then something got it up closer to negative 55. That means it was somehow sodium channels were opened on that cell body and let positive sodium in. So it kind of made it said negative 70, negative 68, negative 67. Something is, so there's a leakage there because there's been tickling or exciting by these other neurons. They're letting sodium in and they're making the cell body get less negative. And if it hits that threshold, negative 55, that trigger, it's going to fire. Sodium channels open, making positivity inside the cell. Then sodium channels close and potassium channels open. Potassium leaves and it makes it more negative again. It actually hyperpolarizes it, brings it super low, then it comes back to the negative 70 again. Yeah, I want to get to this. Nothing. So this just shows you at top is the action potential. Below shows you what's happening to the sodium and potassium channels. So sodium channels are going to open and shut. Potassium channels open and shut. Just so it kind of shows you what's going on. And there's got to be that delay or it doesn't work. You guys are very quiet. That's good. But let's go ahead and take a little 4-minute break here and digest this. And you guys can ask me a question when we come back too. So 8:45, we'll start again. Got it, Jay? Multiple choice. It's not going to be a long one. You know what I mean? Well, you guys, answer any questions you thought about? Hope you're getting this. It's all about sodium potassium channels, and these things are really cool. They're really fast. They just open and close, like really, really fast. And it's about electricity. And the cell body itself of the neuron is graded. That can go it can go from negative, it says a negative 70 usually, negative 70, right? But it can go negative 71, negative 75, negative 80, it can go negative 60, it goes all over the place. But the axon that leaves is all or nothing. It either fires or it doesn't. So that's the big difference here, is that the body is going, it's listening, it's to excitatory, inhibitory. It goes back and forth, and only if it has negative 55 in this trigger zone, the axon hillock is called, the axon leaves, then it begins this action potential. And the action potential is going to be propagated all the way down to the end. So if you have a signal way up here in your lumbar region, spinal cord, and it tells your calf muscle to go, it's going to go all the way down past your thigh, down to your calf, and it's going to branch that axon to all the different motor fibers, and it's going to make my soleus or gastrocnemius, the motor units contract. So putting it in context. All right, so the action potential is all or nothing. It either fires or it doesn't. But the cell body is graded. It goes up and down. You can go up and down, but that can only go to negative 55 and it fires. If it doesn't get there, it does nothing. Yeah. And so this kind of an animation just showing you, it happens kind of quickly, but again, it's just sodium opens, channels closes, potassium channels open and close with a little bit of delay. Then we're going to see there's a little bit of time after that where you can't excite it yet. There's this little refractory period. We'll talk about that. And that's necessary. It keeps the signal moving down in one direction. So it's going to just travel down the axon in one direction. And we look at speed. We can measure this in lab. We can measure how fast it goes down your median nerve by exciting your nerve in different spaces, and on the computer see how long it takes the signal to reach your muscle. And usually in lab it's like 50 meters per second, 60 meters per second, which is pretty freaking fast, right? It can go up to 100 meters per second. Yeah, imagine going across more than a football field in a second. That's how fast this impulse can go. And with your reflexes, that's true, right? You step on something sharp, it's really kind of quick that you react, right? Yep. But when it depolarizes, it excites the membrane next to it, and it just travels down. And it's only going one direction, you're going to see, because these channels, they shut down for a while after they fire. They can't go for a little bit. All right, I show you this, and I got this on the internet. I thought, it looks kind of cool. And I looked at it carefully, I'm like, something's wrong. So I said, well, I'm going to show it in case you guys thinking and such. But this isn't traveling, and this is on a myelinated. This is when if you haven't seen the first lecture, that you don't know this, these are going to be where the Schwann cells, or let's leave it at that, and is going to insulate it. And so the signal is going to jump like here, like this. And the deal is here, it shows it getting less and less, but that's not the case at all in real life. In real life, it's just going to travel down without losing any of its oomph. If you yelled, or like you pass a signal on between people, like that telephone game, the signal changes as you go along. In this case, it doesn't. It's traveled faithfully all the way down. Oh no, let's see. No, I don't want that anymore. Options. Automatic. Oh, hell yeah. All right. Is it working? All right, I was going to show it as it goes down. And you're going to have these are the sodium potassium channels. Oh, look at that. So the sodium channel open. Those red things are sodium. It's going to rush in. And then it's going to excite the next one. It's going to open these sodium channels. And the potassium channels are opening back here, and they're like repolarizing it back to normal. And so that's how it's propagated down. And because it's insulated, see this area here where nothing happens? It's going to be quicker because that signal jumps. Called saltatory conduction. Saltatory is jumping. And it's going to jump down. That's how it's propagated down. And it's faster because it doesn't have to slowly go down the whole membrane. It can jump between these gaps. So that really shows you nicely. All right, once again, this is a question to be on the test. Something that you need to know is that it's all or none. It's action potential. It's not little action potentials and big action potentials. It's like telling you, is it a hot pan or is it a warm pan? It's not bigger and smaller action potentials. And then amazingly, I talk a lot about this in the first lecture, is that what reaches my brain is just one of these blips. So action potential, always the same size. Now I can vary the signal by sending a lot of signals, like 50 per second versus 1 per second. So I can vary that by how many I send, but it's the same size signal. And this little blip, this action potential, is the same thing my brain receives, whether I'm tasting a piece of chocolate cake, whether I'm listening to music, whether I'm being stabbed with a knife. It's the same action potential that reaches my brain. And it just depends on what part of my brain is reached that I perceive it or I interpret it as being chocolate cake versus the smell of garbage. All these things have the same freaking signal. It's just an action potential. That just is going to blow your mind. You guys realize your entire sensory experience of the world is all broken down to an action potential. This stupid thing. And your brain is so cool and so hardwired that which part is stimulated is your brain tells you what that means. That's the difference between being stabbed and enjoying a bagel, that kind of thing. So anyway, as you get into that, it's really cool. And that's all we are, is blips of electricity in our brain. It gives us everything from pain to pleasure. It's so cool. All right, so did I get it clear? Same strength no matter what. It's not big and little action potential. It's either fires or not. Well, here's this resting period I talked about. It's actually kind of important that once an action potential happens, there's a refractory period where those sodium channels are shut for business. It's really short, but there's an important little period. So both channels, the refractory period, the absolute refractory period is, I don't care how much you prod it, nothing's going to happen. And then after that, then it can fire again. And there's a relative one after that, it's just a little minor thing, but where if you really super stimulate it. So, looking at this timing, does it tell you the timing? Yeah, 1/2500th of a second. That's the absolute refractory period. And if you do the math, theoretically, you could have 700 impulses per second with this little resting period. But in reality, it's never more than 100. Still, that's impressive, 100 per second, right? Not per minute, per second. That's how many signal action potentials can happen. You know what's happening? That those channels are opening and closing. There's a change in the charge. All that happens 100 times in a second. It can happen. The fastest. And this will show you on a chart that you can see that once the channel is open, there's this refractory period where nothing can happen. But over here, it can go again. So you have to have that. And that little resting period allows the signal to go one direction. It can't go back because these are resting. The charge has already gone down. So it kind of keeps things going in one direction too. All right, so refractory period is a time when the channels are shut down for business. And after that period, they can be stimulated again. They're asking you how many times per second can it fire. You guys would say about 100. All right, well, you guys have fast nerves and slow nerves and everything in between. You may say to yourself, in evolution, why aren't all my nerves the maximum speed? Make them all big and myelinated. Well, evolutionarily, if you make kids that are over-designed, they've got like 3 kidneys, they've got 2 hearts, they've got all the best nerves, you're going to be out-competed by the moms having cheaper babies. I know it sounds weird, but you are. So you don't over-design an organism for more than it needs to. Just like if you buy a hot water heater, you usually have a 10-year warranty. They usually go bad like year 11. You guys are not old enough. It happens. And they designed the water heater to last that long. And if you do it any longer, it's a more expensive water heater. So it's the same thing with evolution of people. And so that's why I'm saying you have some slow nerves for maybe like, oh, what does my belly feel like? You know, how's my uterus going? You know, and you have fast nerves for like, oh, I'm stepping on a piece of glass. That's got to be fast. So the nerves, the motor nerves going to your muscles are fast. Those going for, oh, how cold is it? And what's my guts doing? Those are slower fibers. And you make them smaller, they don't have to be insulated. So that's the key, is that, does it show it here? The thicker the axon is, the faster it is. So the bigger the axon, thicker, the diameter, the faster it is. I'll show you data. And then the second variable is whether it's myelinated or not. And I went over this in the lecture that's online, you guys can see that. But if it's myelinated, it means it's insulated. It has those gaps, those little nodes of Ranvier in it. So that tells me how fast an axon is how big it is, and is it insulated or not. If it is insulated, it goes faster because it can jump. Look at some data here. Look at this big axon. It's going 100 meters a second. Look at this little one. It's going half a meter a second. You see the difference? Big difference. All right, you guys got a mixture in your body. Like I said, it just depends on how much speed you need. Now, here's some data for you. Look at that chart. If you like data, you like data. You can see this is the size of the diameter. The speed goes up big time, the bigger the diameter. I mentioned that squid has giant axons. Well, like a lobster too, because it's just ready to go. If you touch it, the tail just goes boom to allow it to quickly escape. You know? So things like reflexes like that, you want big diameter axons. And down here in the darker line, unmyelinated, oh, look how slow it is. It never goes about 10 miles an hour, 10 meters a second, see? So you got to insulate it, and you want a bigger one if you want speed, which can be life or death if it's a reflex, right? All right, beautiful. So this jumping is called saltatory conduction. And I study frogs, some of you may know that. And we talk about they have saltatory locomotion, like a rabbit. They leap. This is leaping. And it's a way to speed things up. Instead of it just slowly propagating down the axon, it jumps. Now, it can't do anything between these gaps because this insulation is fatty. It's this myelin fat. And water and oil don't mix, right? And so you're not going to have any like sodium potassium moving there because it's just stuck around this fat. But these gaps are where it happens. It's watery by these gaps, and that's where the thing happens. So the signal jumps. It goes really fast. And this is unmyelinated. And if you see my lecture again, you'll see it's not truly naked. It's kind of like surrounded by a Schwann cell, but it's not wrapped up. It's just kind of like in a little groove. But you can see it's slower. It has to go on the whole damn length. The whole thing has to be polarized. If it's myelinated, just little segments. All right, pretty clear, right? So myelinated is going to speed things up. Unmyelinated, it's going to just propagate down. Beautiful. All right, a few other things I want to talk about here. We asked about muscle cramps before. Often you say we need more potassium. So for muscles and for nerves, you have to have the right electrolytes. It's all got to be right. You have to have, obviously, sodium and potassium. Those are the big ones, sodium and potassium. And you can see plants give you a bigger sodium they have a lot of potassium in them. So banana, spinach, so that potassium, is important and you get those from plants especially. And you can imagine if you have too much potassium out there, and in the lethal injection cocktail I used to kill people, potassium chloride is like the last step. And this is going to stop the heart. So if you have too much potassium out there, you can imagine that, it's supposed to be not much potassium out there. But if there is going to be too much potassium out there, there's not going to be that big of a difference. And the nerve's going to be really excitable. And it's just going to fire all the time. And then if you have not that much potassium, it's going to be not able to fire. It's going to be like so polarized, it's going to be like locked. So it's not going to paralyze. So potassium levels have to be correct. And too much potassium will stop your heart, stop your nervous system. And too few as well. That's why your body has all these ways that it keeps homeostasis. So your sodium, potassium, calcium, all these things are kept at a nice level. If you're in the hospital and your potassium levels are too high, they jump on it, give you drugs to bring it down, you know, or either way. So you got to look at sodium, potassium, and you look at someone's readout to make sure they're okay. So it could be a kidney problem, you're peeing out too much potassium or not. Could be a dietary problem. All right, so they have to be correct inside and out in that fluid for it to work right. Oh yeah, so how does anesthesia, like you put a little bit of a Novocaine, or what's that? What do you put on your skin to deaden it? No, what would they give you if they wanted to like deaden a piece of your skin? What is that? Lidocaine? Yeah, all these canes. And what they do, believe it or not, this is so cool. They screw up the sodium channels so they can't open. You stop the sodium channels, the nerve can't fire, and so the pain signals can't be fired. I don't know if there's there. I guess you don't feel anything too. So if you stop the sodium channels and you just lock them up, you're not going to feel any pain because those nerves aren't going to be sending anything to your brain, right? So that's kind of cool. You can fool those sodium channels. Luckily, it's temporary, so you're not like forever lost feeling in the part of your body. All right. I think I've done with talking about sending that action potential down the axon. And again, it can go all the way from your butt region to your toes, all the way down that huge nerve, down the axon. It's propagated down. Because it's got that kind of speed, it's okay. I mean, reflexes can work. All right, so we're looking at whether it fires or not. So we're talking about the cell in question, the cell body of this neuron, and whether it fires or not. And you see postsynaptic potential. We're talking about excitatory and inhibitory postsynaptic potential. Now, what does that mean, postsynaptic? Well, it means we're talking about the nerve in question because the synapse goes to the nerve on the dendrites or on the body. So other nerves come in and they're going to have a synapse either on the dendrite or right on the body. There could be thousands and thousands of them. And they're going to control that neuron in question. They're going to cause the polarity, how negative it is, to go up and down. If it's excited, it's going to go more towards zero, depolarized. If it gets inhibited, it's going to be more negative, like negative 80, and it's be less likely to fire. So that cell in question adds it up. It's called summation. You're going to add up all of these signals. It doesn't like do it on a calculator, do it on its fingers. It's just chemicals, and it's just the charge. And we're going to see if it's going to hit negative 55 or not. So summation, called summation. It's going to add up. Remember, I was talking about your friends, some of you encouraging you, some of you stopping you from doing something. Same thing. You're going to have excitatory and inhibitory signals. Let me ask you guys to think about this. What would happen in one of these neurons that is telling it to inhibit it, that tells it to not fire? What kind of channels would open up in order to make it not fire? Anyone guess? Like what channel would you open up to make it more negative? Not sodium, right? You guys have a guess what kind of ions you could let in to make it hyperpolarized or less negative? More negative, I mean? Yes. Potassium or sodium, both positive. So if you let positive in, it's going to depolarize, bring it closer to that trigger. What ion did you let in that would make it more negative? Chlorine. Yeah, chlorine. So that's one case. Open chlorine ions. More negative comes in. Instead of being negative 70, now it's negative 75. And so this neuron is less likely to fire, right? Yeah, it's been like held back to negative 75. So it takes even more to excite it. So that's what happens. Those inhibitory ones, they let in negative ions. And so they're going to hyperpolarize it. And the ones that are saying, do it, they're going to be letting in positive ions to make it closer to that threshold. Yep. And it's going to sum both in space and time. And so, you know, if you have signals all at once coming that are going to be excitatory, that's going to be better than the staggered. And if they're closer to the hillock, it's going to have more effect if they're further away. But the cell in question is going to mentally sum up all of the charges, negative and positive. It just happens, right? And it's going to sum them up, and it's going to fire or not. All right, that's all the choices it has. It doesn't have a half fire. It doesn't have a whisper. It either fires or not, action potential or not. And only if it hits that trigger, negative 55. Yeah, I like it. So you can see here that, see all this red, it's getting excited by these guys. These guys say, oh my God, that's a bear. We got to run. We got to run. These guys are saying, ah, it's a black rock. It's a rock. It's February. There's no bears out there hibernating. But he's like, it's a bear. And if enough of them say it, it's gonna hit that negative 55. It's gonna fire. It's gonna tell you to run. All right, synapse. I talked about this in the online lecture a little bit, but that's where the rubber meets the road. That's where one neuron is going to talk to another, or a neuron's going to talk to a muscle. We did that last time. Yeah, and so this is showing up close. This is the neuron in question I'm talking about. And here's all these other neurons talking to it. And so there's synapses there, and some of these will be excitatory. They'll open up sodium channels to make it more positive in the cell. Some will be inhibitory. They'll let in more negative, so it's going to hyperpolarize it. But all of these are changing the charge of this neuron in question by either opening positive or negative channels. Yeah, cool. And so you'll have, again, some neurotransmitters will be excitatory, some will be inhibitory. Okay. Yeah. Some words. And here you go. I've seen this in a test question too, the EPSP, right? Inhibitory or excitatory postsynaptic potential. So as I said, it's green light or red light. I kind of put that on there. And just make sure you understand, if you're going to be inhibitory, you're going to hyperpolarize that cell. You're going to hyperpolarize it, make it like negative 100. That way it's unlikely to fire because it has to hit that trigger, negative 55. If you want to excite it, you're going to depolarize. You're going to make it more towards the zero, so towards negative 55. All right, and so the cell summation, it's going to sum up the inhibitory and excitatory and see if it hits that trigger, negative 55. Cool. And I told you that the ones close to the axon hillock, they have a little bit of a louder voice. So I'll just add that in there so there can be some little fine tuning like that. Some friends are more important in their advice than other friends, this kind of thing. All right, so neurotransmitters, the only one you guys know that I taught you is acetylcholine, and that's the most important one. That's found in all the muscle junctions, acetylcholine. It's also found in the brain. But in the brain, there's a whole bunch of other ones. And there's actually, you get into detail 100 different neurotransmitters. Oh my God. So if you look at that list, you guys recognize some of those. Dopamine, serotonin, as mood altering. You've heard of those things. Even histamine, which we talked about. I think we talked about blood vessels. GABA, oh yeah. And epinephrine. So these all, they're all familiar. To me, just tangentially to different kinds of neurotransmitters. So there's lots of them. Acetylcholine is always used in the muscles. And so what happens, I think you guys know, I can draw you in here. Here's the end of a. So what happens is that the signal comes down. This is the action potential. And when it comes to the end of an axon, it's going to open up calcium channels, and calcium is going to come in. So that they're electrically gated. So when the electricity reaches, calcium ions open. The calcium is more of it out here. It's going to come in, and that's what's going to start making these little vesicles or these little bags filled with neurotransmitters, some kind of chemical. They're going to bind to the membrane and they're going to dump it into the cleft. Yep. And so this membrane of this vesicle is going to become part of the membrane of the cell. And then on the other hand, on the postsynaptic neuron or muscle over here, there's going to be receptors like that. And this neurotransmitter, sometimes takes 1, sometimes 2, is going to bind with these receptors, and they're going to open up channels. Like doors are going to open up, and then usually sodium is going to rush in. And that's going to potentially excite the next one. We just talked about this. Or maybe it's going to inhibit. Maybe this neurotransmitter is going to open up chlorine. It's going to be more negative ions in it. That's what happens. Another important thing to know is that neurotransmitter in that little synaptic cleft, that little space, has got to be destroyed and recycled. And there are drugs that prevent that. And then you end up with a super excitable muscle, you get convulsions. So you have to be able to destroy it and recycle it, or else it continuously fires. And then drugs can get in there that are similar to the neurotransmitter, and they're going to bind with the receptors. So you can give that person that drug, and they're going to be super excitable, or it binds up the receptors but doesn't open the channels, and you've deadened it. All of a sudden, you've made it useless. So to show you how drugs can work, because of the chemistry in this synapse, because you can fool with these chemicals. All right, and so hopefully the signal goes, the neurotransmitter is destroyed, and then if another signal comes, it happens again and again and again. The neurotransmitter is usually recycled and used over and over. Okay. You guys got the basic synapse? And remember, that neuron in question can have 10,000 synapses, 10,000 neurons talking to it. All right, one more thing just to let you know, in the brain, and then we look at brain chemistry, you can have other chemicals in there, neuromodulators or neuropeptides that can modulate the signal. You can either make it more likely to fire or less likely to fire. And some things you should be familiar with are endorphins. If you work out, even tanning causes endorphins. It's called endogenous morphines. It makes you feel good. It goes down to your brain. And caprolins are the same thing. And they'll happen if you're really injured or have a lot of stress. You'll give this off to help soften the blow of being so in pain. And substance P causes you to be in pain. In other words, the caprolins and endorphins, they will inhibit substance P that causes pain. And lastly, I want to talk to you a little bit about addiction. All right, don't do drugs, kids. All right, is that opiates were discovered, and oh my God, thousands of years ago, like 4,000 years ago, a long time ago, opiates were discovered. Poppies, they had to give off this chemical, and if you smoke it or inject it, or it causes you to feel euphoria. It deadens pain, and if you use it outside of pain use, you know, you get high kind of thing. And it turns out that they're the same kind of chemical is very similar to our natural endorphins. Is this morphine from this plant? Now the plant doesn't do it for us, but it happens to be similar enough that it works for us. We figured it out, and so all the opiates, all the opiate problems, because you can have tremendous addiction. And addiction has been around since the Pharaoh days, to opiate, things like that. And what happens is it changes your brain chemistry. It changes the receptors. And it can happen in many different ways. What often happens is that the hallmark of addiction is it takes more drug to make you feel better, like nicotine. Like you start with one cigarette, and then you need a pack a day. And because it actually changes the receptors, and so that you need more drug, because your body adapts to that. So the withdrawal symptoms are, if you take opiates, your body makes less of the natural endorphins. And so when you cut cold turkey, you feel like horrible. You feel terrible. And so until your body can kind of go through that withdrawal symptoms. So that's the deal. You take it away from your body makes less of the natural stuff, and it's been used to what you were giving. And nicotine, of course, horribly addictive. Well, my parents smoked, I had a girlfriend that smoked. It's so difficult to quit, apparently. But nicotine will bind with some receptors in your brain that gives off these pleasurable feelings. And if you don't smoke, you crave it. You're missing this nicotine. And horrible addiction. It's from a tobacco plant. It's a toxin. It's used to so the bugs don't eat it, they die. But we've learned that it gives you this kind of a feeling in your brain, but it changes your brain chemistry. It keeps you from recycling these receptors, and you need more and more. Yeah. All right. I think what time is it? Ain't about time. I guess last thing was, when we looked at these, when you look at the circuits in your brain, I want you to realize that you have converging circuits. Like Is it a bear? So I have like my memory and I have like my vision. And then diverging is a way for one neuron to talk to several. And then there's even like ones that are circular too. But that's how we get to all these processes in our mind, are these circuits between our neurons that develop. All right, you guys. Woo, that's chapter 10, believe it or not. Watch my online one at home, and you'll be set.